Have you ever thought about what all goes into creating drugs and treatments for diseases? One important concept that is studied is force. Let's take the example of people pushing an object. For case 1, the object won't move because the two people are pushing with an equal force. In case 2, the object will move to the right because there are more people on the left pushing. Hence, there is greater force on the left side. In case 3, the object will move to the left because there is a greater force on the right. Force is everywhere, including between molecules. Two molecules are constantly creating a force, which is important when making drugs or treatments that target those molecules. This is a typical microscope that is used to study force. However, machines like this one are very expensive. That's why, at the Nano Engineering and Biodesign Lab, we created an extremely small structure called the RLS that can help us study force between two molecules. The structure shown here is made using DNA. DNA is found in every one of our cells. It is the key to our cells functioning properly. DNA is a two-stranded structure made up of base pairs A, T, C, and G. The 4 base pairs always match up the same way. A will always match to T and C to G. In this example, blue is always matched to yellow and red to green. We use this concept to make our three-dimensional structures in the lab. We take a long, circular, single strand of DNA and add something called staples. Staples are short strands of DNA containing the bases that we just talked about. Those bases then match up with bases in the circular strand of DNA. Eventually, once enough staples are added, along with heat, we will get our structures. The RLS structure can be seen here. As shown in this gray image, our structures are very small, and we can also create a lot of these structures. But wait, how does this structure measure force? Well, DNA can also act like a spring. Let's take a Slinky for example. It can move up and down, side to side, and be stretched or pushed together. While all these movements are happening, there is a force in the Slinky. These are all things that DNA does too. That's why we use a separate strand of DNA in the RLS. When two molecules come together, the DNA, like a Slinky, will stretch. Likewise, when the molecules pull apart, the DNA will ball up. And that is how we use the RLS to measure the force between two molecules. Now, can you use all these concepts to make your own structure to measure force between molecules?